I would like to present my journal club topic about chitosan-based scaffolds and their applications in wound healing. Uh, in this journal is published in Elsevier and published by the authors Shakil Ahmad and Saika Igram, published on 2016 April 7th. This is the picture of the chitosan, structure of chitosan. In this picture, the first position and the fourth position is linked with linked in the glucose linkage. This is the 1,4 glucosamine linkage. It is a biopolymer. This we can see the one by one in the upcoming slides. So the contents I will explain in first for purpose of the selection and introduction, biomedical applications of chitosan, possibilities of processing chitosan into different formulations, and chitosan based materials for wound healing. In this first purpose of the selection, why I am selecting particularly for this article in the sense because of the chitosan is the only polymer I have selected for my project. Because of the chitosan have a wide variety of advantages, which is a cationic biopolymer composed of glucosamine linkage. The one, two, or four linkage is connected, and this is a non-toxic, biodegradable, more compatible with the other selected synthetic and natural semi-synthetic polymers, and which is cheaply available and have the variety of applications in antimicrobials and inflammatory, disinfectant, and wound healing, which is able to formulate any other formulations like a membrane, nanogels, nanofibers, and nanosponges, etc. So only I have selected for the chitosan as a polymer for my project. It's the introduction. What is first of all, many of the researchers are currently choosing the polymer-based dressings for the dermal applications of wound healing, whatever in the sense. Uh, Many of the but some of the synthetic polymers have produced some of unwanted effect or irritants are happen. This is not suitable for all. But in the biopolymers, they are collected from the polysaccharides such as cellulose, starch, a carbohydrate as well as. And this is the chitosan is obtained from the animal sources, and the biopolymers are obtained from the bacteria, fungi, and animal proteins based polymers. By the way, in this article, we will briefly know about the chitosan. This is the chitosan. Chitosan is this is the structure of chitosan. In this chitosan is consisting of first position and the fourth position is linked 2 acid amido, 2 deoxy, 2 glucopyranose, and 2 amino, 2 deoxy, 2 glycopyranose units. And this is generally obtained from the chitin. The chitin is obtained from the various animal sources, plant, like I will explain in the upcoming slides. And this chitin is undergone the deacetylation process, which is from the chitosan. And the chitosan is a biocationic polymer having the pH 6, so which can readily interact with negative charged molecules such as proteins, anionic polysaccharides, example alginate and carrageenan, fatty acid, bile acids, and phospholipids. And uh, uh, the biopolymer nature of the chitosan, so only which is biodegradable, non-toxic, and antibacterial effect and biocompatible with when we blend with the other polymers or the natural substances which can biocompatible and which can able to form the many of the formulations like a membrane, nanogels, and nanosponges, etc. This is the sources of chitosan: Aspergillus niger, squid pen, robust waste, ground waste, crab waste, soldier fly, and larva mushrooms. From these sources, chitin is obtained. From this chitin, undergone the deacetylation process only, we obtain the chitosan. the modification of the chitosan. This is like a prodrug. For example, if for a prodrug is used for the enhancing of the taste or enhancing of the absorption of the drug. By the way, like that, the chitosan is the polymer is needed to modifications for enhancing the actions or promote the adhesion, like and all. By the way, chitosan is modified with the coating, oxidation, lower temperature plasma, and surfactant addition. In order to chitosan is blended with the various additives. So after the addition of the additives, which can evaluation is necessary because of the biocompatibility is not all the this is not related to all the polymers and natural products. So evaluation is necessary. Therefore, the various biomedical grade chitosan derivatives is necessary to engineer model. That is a high quality of biocompatible for the human wound management. And the most advantages of the chitosan is we can made into a gel, membrane, nanofibers, nanofibrils, beads, microparticles, nanoparticles, etc. And these are the possibilities of processing of chitosan into different formulations: membranes, nanogels, films, nanofibers, hydrogels, nanoparticles, and beads, and the nanosponges also. Biomedical applications of chitosan. In this Zhuo and Teodo in 2016 and 2014 they were reviewed. The chitosan is the best polymer for the biomedical application and for the different ways. Chitosan is used for the disinfectant also and the drug delivery, the gene delivery, bioimaging, hemodialysis, bioadhesives, anti-diabetic, wound healing, and tissue engineering. In the antimicrobial nature of chitosan, antimicrobial agents, in the sense they are the agents that kill the microorganism or they inhibit the growth. That is called the chitosan. By the way, the chitosan is the best cationic polymer, so which can readily interact with the gram-negative bacteria and decrease their acute toxicity. When the chitosan is added with niacin, is the antimicrobial agent. There's an there no need to be added the chelating agents because of the chitosan is also act as a chelating agents. Um, in many of the wound healings, example burn wound, in the sense the affected area get microbialized. So we have to apply the microbialized and become a sepsis. So we have to prevent the sepsis and healing the wound. In that way, the chitosan is play a wide variety of role in that wound healing, the type of wound healing. The anti-inflammatory property of chitosan. In this, Wakamoto in 2002, they were reviewed. They were induced anti-inflammation, with the 0.5 percentage of the acetic acid solution, and this prepared and this produced inflammation is reduced with the help of the chitosan by the administration of the chitosan, which can readily reduce the bradykinin and other inflammatory mediators. And N-acetyl glucosamine, it's an anti-inflammatory drug which is synthesized in the human body from the glucose and which is incorporated into the glycosamine glycoprotein and glycoproteins, chitooligosaccharide, etc. So which can molecular weight of 5 kDa. In that incorporated substance is very powerful than the non-steroidal drug of the indomethacin. This can reviewed by Kim and their groups. The chitosan-based material for wound healing. This is the chitosan in wound healing alone or merged with the other polymers or the natural products. Chitosan with the synthetic or natural polymer blend scaffolds in wound healings. Chitosan-based composite scaffolds in wound healings. Chitosan based sponges in wound healing. Chitosan based oil immobilized scaffolds in wound healing. Chitosan based extract immobilized scaffolds in wound healing. Chitosan based drug loaded scaffolds in wound healing. First, chitosan in wound healing. In that, the chitosan and polyvinyl alcohol prepare the nanofibrous, and it is the promoting the L929 cell attachments and proliferations, which can reviewed by the Zoe and their team in 2018. And the chitosan is the biopolymer, so which can non-allergic, non-toxic, and keep the environment in the moisture conditions. And chitosan is not only killing the microorganisms, which can promote the wound healing also by acting on the hemostasis process, by acting on the hemostasis phase and promoting the wound healing. In this, the composite sponge of curcumin, chitosan, and gelatin of the nano sponge is prepared, and their formulation as and the formation of the sponge is and the composite sponge were from the enhancing of the formation of collagen and wound closure, and the preparation is improve the wound healing activity. And chitosan with synthetic and natural polymers blend scaffolds in wound healing. In this, chitosan grafted polyacrylic acid was prepared, and polyacrylic acid of the hydrogel is prepared. And this hydrogel is normally after 14 days of the culture by the chitosan hydrogel is suitable for the wound healing, which can not degradable under the period of the 14 days. And in the Ribeiro and their groups in 2009, they were reviewed. They were isolate rat skin, rat fibroblastic cells from their rat skin, and they apply the hydrogel for chitosan hydrogel and promote the cell adhesion, and they will report the cell the chitosan hydrogel as the promote the cell adhesion and the proliferations. And the biocompatible chitosan polyethylene glycol diacrylate blend with they will make the film, and these are suitable for the material show on cytotoxicity and the growth of the they were towards the growth of the L929 cell, and had a good in vitro biocompatibility, so it can use for the wound dressing material. From the previous slides, we can confirm the chitosan is compatible with the synthetic polymers as well as the natural products also. The chitosan based composite scaffolds in wound healing. In that study, chitosan and a serine composite of nanofibers are prepared by electron spinning method. In this method, in the x-axis, concentration of the composite nanofiber in the microgram per ml, and in the y-axis, cell viability, the percentage of control of the cell viability. The prepared chitosan and serine composite of nanofiber have the diameter between 240 to 380 nanometer, and which can antibactericidal activity against the both of gram positive and gram negative. In this, the gram negative or the gram positive cell viability, which can the more cell viability control. In this diagram, 24, 48, and 72 hours respectively, 16, 32, 62, and up to 250 microgram per ml concentration is used. In this study, after this 72 hours, have the more above the 100% of the cell viability control compared than the 24 hours and 48 hours. From this study, we will conclude the chitosan is the best antimicrobial agents, and they will control the cell viability of the gram negative and gram positive bacteria. Again, in the different concentrations. In this study, FE-some images of the hydroxyapatite polymer and chitosan silver hydroxyapatite. In this, A and B respectively hydroxyapatite polymer, and the C and respectively chitosan silver loaded hydroxyapatite polymer. In this two, in this B and D shows the very needle-like and needle-like crystal is observed in the both the HAP and the CS silver HAP coatings. They were showed they are promising the material with the biomedical applications. The chitosan based sponges in wound healing. Sponges are the new material in the biomedical formulations. Um, uh, why? Because of the sponges is chosen. Because of sponges have the many fluid absorption capacity, and they release the drugs in the controlled release manner, and they have the wide variety of properties in it. In this sponges based chitosan is explained by Meade and their groups. They were explained. They are separated into three groups. One is the cotton gas, the second one is the chitosan and alginate based sponge, and their third one is chitosan, alginate and curcumin based sponges. In these prepared sponges, the chitosan, alginate, curcumin loaded sponge is a very provide the wound healing in more wound healing compared than the chitosan alginate as well as in the cotton. The sponge-like dressing based chitosan, glutamate and sericin was developed for the treatment of chronic skin ulcers. And moreover, the optimized dressing is able to improve the fibroblast proliferations and promote the wound healing. The chitosan based oil immobilized scaffolds in wound healing. The castor oil and chitosan modified into zinc oxide nanoparticles. Chitosan that can make into be a film. In this prepared film is effective against the antimicrobial activity against both gram-negative and gram-positive. It depend upon the concentration gradients. The chitosan-based extraction immobilized scaffolds in wound healing. In many of the formulations, some of the plant sources directly apply onto the wound. But that while we directly apply onto the wound is not curing properly, or the healing process is could not enhance it compared than the polymer based drug delivery or polymer based wound healing. By the way, hydrogel film of encapsulated with gentamicin sulfate, salix alpha, and Juclina regia leaves extract showed the both antibacterial and antifungal activities in disc diffusion method and in the minimum inhibitory concentrations method. The wound are and if your wound is happen in a particular area, the microbial contamination will be there. So we have to reduce the microbial contaminants. By the way, the aloe vera, the natural substances or the other antimicrobial agents are needed to be cure that wound. This is the zone of incubation of first picture is the chitosan alone and the chitosan aloe vera composite. Chitosan aloe vera composite in 1:1 ratio. In this the third picture, chitosan aloe vera 1:1 ratio showed the more zone of incubation compared than the chitosan aloe vera and the chitosan alone. This is the HDFBS cell study, human dermal fibroblast study. In this study, the chitosan aloe vera and the chitosan aloe vera 1:1 ratio membranes are taken in the cultured 1 to 7th day. In that membrane, we have to seed onto the HTF cells on the membranes of the chitosan aloe vera and the chitosan aloe vera 1:1 ratio and observe the study. In that study, the chitosan and aloe vera 1:1 ratio shows the reduce the cell proliferations and cell spreading compared than the chitosan and aloe vera. The chitosan based drug loaded scaffolds in wound healing. The thiolated chitosan with poly and isopropyl acrylamide loaded with ciprofloxacin, which is for the suitable for the productive wound for a prolonged period. In this combination of the thiolated chitosan with poly and isopropyl acrylamide and ciprofloxacin show the antibacterial properties to the well-known bacteria of E. coli. And the conclusion of this journal, in this polymer of chitosan have a wide variety of properties such as antimicrobial and inflammatory and other biomedical applications. And in the wound healing, they have the wide variety of they are available in the wide variety of formulations like a membrane, nanogels, and nanofilms, etc. In the future, this chitosan polymer is the best polymer in the dermal applications. And these are the references I have referred for the preparation of this article. Thank you.